Every human life begins with a single, remarkable event.
In this video, we'll follow that journey, from sperm meeting an egg, to the formation of a growing embryo.
And even into one of biology's most fascinating variations, identical twins. When sperm enters the female reproductive system, it travels toward the fallopian tubes in search of an egg.
This marks the beginning of potential conception.
Once a sperm cell reaches the egg, it must penetrate the egg's outer layer. Only one sperm can break through PACE. The moment it does, the egg seals itself off from all others.
As the sperm enters, its tail is no longer needed and disintegrates. The head of the sperm fuses with the egg, and the two nuclei begin to merge. Together, their genetic material combines to form a complete set of 46 chromosomes.
Half contributed by each parent.
This newly formed single cell is called a zygote, dengote, the very first cell of a developing embryo. Remarkably, it already contains all the genetic instructions needed to build an entire human being.
The zygote quickly begins dividing, first into 2 cells, then 4, then 8, forming a small, growing cluster. Gentle muscle contractions and tiny hair-like structures called cilia guide it toward the uterus.
This journey typically takes 3 to 5 days. By the time it reaches 16 to 32 cells, this structure is now called a morula.
Around day 4, the morula begins absorbing fluid, forming a cavity inside it.
It now has 2 parts, an outer layer that will form the placenta, and an inner cluster that will become the baby.
The blastocyst then attaches itself to the uterine lining portbosink, a process called implantation. This connection allows the embryo to draw nutrients and oxygen directly from the mother's bloodstream. In rare cases, something unusual happens during these early stages of division. The developing cells split into 2 distinct groups, each carrying identical DNA.
These 2 groups continue developing independently, each eventually forming its own baby.
Scientists still don't fully understand what triggers this split. Genetic and environmental factors may play a role, but it's largely considered random chance.
The timing of this split determines exactly how much the twins will share inside the womb.
The earliest possible separation happens at the 2-cell stage, forming 2 entirely separate zygotes. Each twin develops with its own amniotic sac and placenta clichures, the lowest risk form of identical twin pregnancy. If the split happens slightly later, the twins share a single placenta, but each keeps its own amniotic sac.
Sharing a placenta means the twins also share a blood supply, which can occasionally become imbalanced.
In severe, untreated cases, this imbalance can carry serious risk for both twins.
Modern treatments, including surgical intervention, have significantly improved outcomes over the years. If the split occurs after full implantation, the twins share not only a placenta, but also a single amniotic sac.
This shared space significantly increases the risk of umbilical cord entanglement.
In extremely rare cases, twins don't fully separate FOPRAs, resulting in conjoined twins who may share organs or structures. This condition presents significant medical challenges, and long-term outcomes depend heavily on the organs shared and the care available. Whether a pregnancy results in one baby or two, the underlying story is the same remarkable process. One fertilized egg, dividing and growing, shaping a new life, or, in rare cases, two.